Hello, everyone. My name is Kayluz, and I am a postdoctoral fellow at the Center for Innovation, Canadian Blood Services, in the lab of Dr. Nora Branch. It is my pleasure to share with you some of the results we have in a project devoted to study cellular mechanisms that may be involved in seronegative RBC hemolysis. Hemolysis is a potentially life-threatening complication associated with different diseases such as immune thrombocytopenia, sickle cell disease, autoimmune hemolytic anemia, among others. Causes of hemolysis are diverse. They can be intrinsic, such as RBC defects, or extrinsic of immune or non-immune nature. Currently, it is believed that immune hemolysis is mediated by humoral responses. Therefore, laboratory tests rely on the detection of antibodies bound to the RBCs or present in plasma. Nevertheless, some of these serological investigations result negative, and there is no current explanation to why these patients are suffering hemolysis. So we decided in this project to see if there could be a cellular mechanism involved in the hemolysis in these patients. So then we set out to select a method that would allow us to measure RBC lysis, and for that we compared three different ones: hemoglobin release, LDH release, and radioactive chromium release. Once the method is selected, we would collect blood from the patients and isolate serum, RBCs, and immune cells that will be combined to perform either a cytotoxicity assay or a phagocytosis assay. From the three methods that we included in this study, we could observe that despite this was detectable by all of them, the chromium release assay resulted to be the most sensitive one. And this is a very important step in our project because this would be applied to patients that can be leukopenic, patients that might not be willing to give a lot of blood. So we needed to use the most sensitive method to limit the use of patient material. We also needed a cell model that would allow us to measure RBC lysis. And for that, we went on with an ADCC model in which we would coat the RBCs with an anti-D antibody. Having selected the model and the method, we then tested different cell populations against RBCs for their killing capacity. We tested whole PBMC fraction, isolated NK cells, and isolated monocytes and different effector target ratios. And we could observe a death of the RBCs by all of these cells. However, when we compare the three different subpopulations, we can see that the NK cells are the ones that have the higher cytotoxic potential. And this was a very good news for us because there is just one case report in the literature of a patient suffering from seronegative hemolysis in which a cell is responsible for the lysis of RBCs, and natural killer cells are these responsible cells. Additionally, we decided to include neutrophils in our study because they can also perform ADCC, and also because neutrophils are key role players in a type of autoimmune hemolytic anemia called paroxysmal cold hemoglobinuria. So we could not discard that neutrophils were also playing a role in the case of the seronegative patients that we were investigating. So knowing that we were able to detect cell death by our method, we went on to study different patients with seronegative hemolysis. And very interesting for us, we were able to detect direct killing of RBCs by different immune cell populations. We understand that here we are in a low range of cytotoxicity, but nevertheless, these results encourage us to pursue this project and to try to fine-tune the technique when patient samples are involved, eventually increasing the time of the assay or varying the effector target ratios. We are also interested in seeing if phagocytosis could play a role in this hemolysis. So for that, we use a model of monocyte-derived M1 and M2 macrophages. That's what's developed in our lab. And we used RBCs that were deprived of glucose to increase the expression of phosphatidylserine in their surface. We did that because it has been reported that patients with sickle cell disease have an increased expression of phosphatidylserine in the outer membrane layer of the erythrocytes. So we tried to mimic such scenario, and we could observe that actually when phosphatidylserine was expressed, we had a preferential phagocytosis mediated by M2 macrophages. And even more interested was the fact that we could observe the same phenomenon in patients suffering from sickle cell disease, so red blood cells derived from patients with sickle cell disease. This was also observed in some sepsis patients in which phosphatidylserine has also been reported in the surface of the RBCs. We definitely want to continue investigations in this direction, including different diseases as well, because, for example, autoimmune hemolytic anemia has been reported to show a preferential Th2 profile. And a Th2 profile will induce the polarizations of macrophages towards an M2 phenotype. So we cannot discard that in patients other than sickle cell disease patients, this phagocytosis by M2 macrophages is occurring. And with that, I want to conclude my presentation, and I thank you all for your attention.